Our immune system is always at war with infections on a daily basis. The snipers of this attack force is a complement system. So today we'll talk about the complement system. So what is the system? So it's a system of some proteins which are normally present in the human serum. So generally what happens is this gets activated not by itself but only when there's an antigen and antibody reactions, and this can damage or lyse the cells and has a lot of other biological activities. So this belongs to a triggered enzyme capsules, means it is a cascade system, just like what we had studied in a coagulation cascade. So every step has ability to activate the next step and will slowly provide an amplified response, and it has its own control mechanism. So it can be very precisely regulated as around 5% of normal serum, which is not increased by vaccination or previous infection. Generally, they don't find this complement getting activated to free antigens or antibodies. So it has to be first of all, the antigen antibody has to combine, and the complement will go and attach to the Fc fragment of the immunoglobulins. Okay. Now the binding is more so with the IgM followed by Ig3, IgG1 and IgG2. Now there are three different pathways. So today we'll talk about complement pathway in a animated version to make it little bit more interesting and rememberable. To make it more interesting and also to remember the pathway easily. So next you have the alternate pathway and lectal pathway, which we'll cover in a later stage. These are the most important steps of the complement pathways. That is, first is the initiation, formation of C3 convertase, formation of C5 convertase, and formation of the membrane attack complex. This has 30 different serum proteins. So it has all the complement fractions, the propaganda system, the control proteins, etc. So the most important member is the first one, which is C1. It's composed of three components: C1q, C1r, and C1s. So look at this animation for a short time. We'll try to understand the classical complement pathway. You have an antibody which is always making rounds. There comes an antigen. So obviously there has to be an antigen antibody reaction. Now this antigen antibody, when they have combined, they will activate C1. Now C1 has three components: C1q, C1r, and C1s. The first one to get activated is a C1q. This will go and attach to the immunoglobulin, which will sequentially activate the other two components, which is C1r and C1s. Now the C1s component is the one which is active in causing the next reactions. So this, once this have combined, that is antigen, antibody, as well as the complement C1, it will now activate the next complement protein, which is C4. As soon as it is activated, a small fraction of it goes out, which forms an anaphylatoxin, so which is the C4a component. So the C4b, which is there, will now combine with the C1 to form what is called as a C14b. Now this has to have an amplified response, so it will go and attach with C2 in the presence of magnesium compounds. So that again leads to cleavage of a small component, which is C2b, which goes and out into the bloodstream. The C2a will combine with this existing C14b. So that forms what is called as a C3 convertase. So it is C1, next you had the component C4, next you had component C2. So these three combined forms the C3 convertase. So as the name suggests, now it has to go and attach to C3. So once the attachment is done, a small part of it, the C3a, goes out into the bloodstream as an anaphylatoxin. The remaining part, that is the existing C3 convertase along with C3b, will form the next most important step, which is your C5 convertase. So from here starts the process of attaching to C5. C5 will again break off a small part, which is C5a, again an anaphylatoxin. So this forms the starting of the membrane attack phase. So the complement, which is still now getting activated step by step, is now ready to damage the bacterial membranes. This C5 convertase is now joined with C6 and C7 sequentially, followed by the addition of C8 and C9 components. So finally, what do you have? You have a proper membrane attack complex. Now to recollect, you have the C1, which is followed by C4b, which joins to it, then followed by C2a, then you have C3b, C5b, 6, 7, 8, 9. This whole thing is an active complex as it has a bar on top of it. Alright, so this is the membrane attack complex. Now this complex has the ability to go and form pores along with immunoglobulins into the bacterial cell wall. So this is how the bacterial killing starts. Now let us quickly go through the steps one by one, just for a purpose of revision. So first you have an antigen followed by a antibody. So the antigen and antibody has to combine. So once combined, the C1 component, which has the C1q, C1r and S. So the C1q part will combine with the immunoglobulins in the presence of calcium and will activate C1r and C1s. The C1s will now go and activate the C4. C4 can split up into C4b and C4a. C4a is a anaphylatoxin, so it will go into the bloodstream. The C4b will join with the C1, leading to C14b. This is followed by activation of C2, again C2a and C2b. The C2b is a anaphylatoxin, again it will go back into the bloodstream. The C2a will combine with the C14b, the previous component, form C3 convertase, the first important step. Next, it will be attached with C3. C3 is again split up into C3a and C3b. C3a will go back into bloodstream. C3b along with the previous C3 convertase will form C5 convertase. Then goes the step of C5. C5 again split into C5b and C5a, and C5b will join with C6, C7 first. Then there's a formation of C8 and C9. So finally you have the membrane attack complex which is formed, right? So this leads to antigen killing. Now if you notice here, in all the whole step, except for C2, in all other components, C2a is a small part which goes out as a anaphylatoxin, except in C2, where C2b is a smaller fragment which goes to become the anaphylatoxin to the bloodstream. Now this complement has to be regulated, so you have self-regulatory mechanisms. So you have inhibitors and inactivators. So first we'll look at the inhibitors. So the first step itself, C1, if you have to stop, it is the C1s-INH. So it's a heat labile protein which can also inhibit other esterases like plasmin, kininogen, Hegman's factors. So what will do, it will not allow the cascade to proceed. There's a S protein which is also present in the normal serum, which will bind to the C6, C7 after the formation of the membrane attack phase. So there it will slowly lyse the membrane attack phase itself, so there's no formation of membrane attack complex. Then you have the inactivators. You have the factor I, which controls the C3 activation, which is important for alternate pathway, which we'll see in the next video. Factor H, which joins with factor I and controls C3 activation. Anaphylatoxin inactivator, which will enzymatically degrade C3a, C4a and C5a, which is your anaphylatoxins. C4 binding protein, which will control activity of cell-bound C4b. So what are the effects of this biological activities of this complement system? It can cause immunological membrane damage, amplifies inflammatory response like hypersensitivity. It has a little bit of antiviral activity. It will promote phagocytosis, promote immune adherence also. So what happens if there's a deficiency in the complement system? The most important one is a C1 inhibitor deficiency, which can lead to hereditary angioneurotic edema. So there's episodes of acute-ness angioedema in tissue. It can happen in the respiratory system or alimentary tracts. It can also become fatal if it's in the respiratory system. What happens? There will be uncontrolled breakdown of C4 and C2 because the C1 is not inhibited at all. So this has to be treated with a fresh frozen plasma, which is a C1 inhibitor. And for prophylaxis, you can use epsilon aminocaproic acid. So this is basically about the classical complement pathway. I hope this was short and sweet. Please do like and share my video, subscribe to my channel. In the next video, we'll talk about alternate pathway and lectin pathway. Thank you.